Speakers is Professor Ellen Holmes. And quick introductions. Professor Ellen Holmes is a leading researcher in the field of computational and systems medicine. She is holding a prestigious Australian Research Council's Laureate Fellowships at the Mueang University in Australia, where she is also the director of the Centre for Computational and Systems Medicine and the Deputy Director of the Australian National Phenome Centre, Mueang Universities. Her early research work was specialized in renal toxicologies, and she has since become the pioneer in the development and implementations of metabolic phenotyping in translational clinical paradigms such as the biomarker discovery. Her recent research also focused on the mapping of mammalian response to infectious disease, including the system's effects of the SARS-CoV-2 infections and the long COVID. Professor Ellen Holmes is recognized internationally for her contributions to the field, and she holds several memberships on advisory boards, editorial boards, and research panels. And she was awarded the Royal Societies of Chemistry Interdisciplinary Prize for her work in the interface of medicines and chemistry, and she has been a fellow of the Academy of Medical Science since 2018. With that, please join me in welcoming Professor Ellen Holmes up on the stage. And with the slight change of the talk title, so today Professor Ellen Holmes is going to tell us about the metabolic profiling in population screening and personalized medicine.
Oh. Great. Firstly, thank you very much for the invitation to come to Kaen. It's always a pleasure to be here, and we're really looking forward to establishing a long collaboration with you. So my title is very similar to Jeremy's. It's really a continuation. Jeremy gave you the big vision. I'm going to drill down into a couple of case studies and also point out some things, some pitfalls and things that you should look out for in establishing a phenome center. Okay, so echoing Jeremy's statement that a lot of disease starts in early life, really the William Wordsworth really sort of brought this home, saying the child is the father of the man, and I'd argue it even starts before that in utero, in pregnancy. And Theodore Roosevelt was famous for this quote, which says, old age is like everything else. To make a success of it, you have to start young. So it's very important to look after yourself during the early phases of life. And I'm going to show you some examples of why. So if we think of our lifespan, when we start off as a baby, unless we have some genetic disease, we are more or less similar in terms of health. We're all very healthy. But as we move through life, it depends a bit as to where we're born and to the environment we live in, as to what the diseases are that we will accumulate later in life. And some of the things that influence diseases are what you eat, your gut microbiome, how much exercise you do, which drugs you take, even your education levels, and so forth. So by the time you get to your 60s, if you're born in the UK or Europe, you're looking at a population whose real big diseases are things like neurodegenerative diseases, cardiovascular diseases, and stroke. However, if you're born perhaps in Central America, the example here, then you are more likely or have a higher risk of diseases such as diabetes, TB, skin cancer because of sun exposure, and lung cancer. So as you go through life, population differences matter more. And there's a concept called allostatic load, which was invented by Berthinger and in McEwen. And the idea is that we're all a bit like, our metabolism is a bit like an elastic band. You can stretch it and it'll bounce back. So if you have a trauma, an injury, and you harm the body, it will bounce back to maintain homeostasis. But there's only so long that you can bounce back for, and it's like an elastic band, and eventually the elastic band will snap. And this is called allostatic load. So I want to just look at a few exposures during life that lead to the chronic diseases like cardiovascular disease and neurodegeneration. So as Jeremy said, we have two main approaches to metabolic phenotyping. One is the population screening, where we try to get as many samples from as many people as we can and use the numbers and the statistics to tell us something about disease risk. The other approach is to take one person or two people and follow them very, very carefully in a lot of detail and do a lot of analytical experiments. So if we consider pregnancy, and this is the formation of the infant, there's a lot that happens metabolically during pregnancy. You have an anabolic phase and a catabolic phase, and you have a lot of changes in hormones. You have increased cortisol, for example, stress hormones. You have a lot more pressure on the cardiovascular system. And this, obviously, pregnancy lasts for 9 months, after which your metabolism returns, or does it? So this is a question, does your metabolism return? And this is a study we did with University of New South Wales, Professor Imad El-Omar, and his team have profiled mums during pregnancy from trimester 1 right the way through to birth and beyond, up to 6 months, 1 year post-birth. And you can see, if you follow the colors, during pregnancy, your urinary and your plasma metabolism sort of deviate, they come out from where you start from, you have the baby, and then they go back to somewhere around where you started from. And we can look at the different changes, and this is just showing the lipoproteins measured using NMR, and you get a lot of changes in your triglycerides, for example. I'll come back to that in a minute. But if you compare non-pregnant versus pregnancy in trimester 2, which is still in the anabolic phase of pregnancy, you can see that you can very clearly separate the pregnancy from non-pregnancy. And if you look at the loadings plots, which are the colored plots on the right-hand side, you can see again, it's driven by a lot of triglycerides and also your high-density lipoproteins. And whether you look at healthy pregnancy or pregnancies with complications such as cholestasis or gestational diabetes, you can see the same effect, but a little more magnified if you have a pregnancy complication. And if we just look at BMI and serum triglyceride levels and compare them in between healthy pregnancy in green and women who had gestational diabetes in purple, you can see that even up to P3, which is 12 months after giving birth, some of these parameters are elevated, remain elevated, particularly in the gestational diabetes. So if you have gestational diabetes as a mum, you're much more likely to develop diabetes later on in life. And also the baby is more likely to develop metabolic diseases later on in the child's life too. So if we can understand what these changes are, it could really help us understand what we can do to intervene in gestational diabetes. So this is just showing similar information in a little bit of a different way, but normal pregnancies are in the orange, healthy pregnancy, and your gestational diabetes is in yellow. And these are radar plots of the different lipoprotein parameters. And you can see 1 year postpartum, for example, in triglycerides, the ladies who had gestational diabetes still have very elevated triglyceride levels, and the same for the cholesterol levels. And basically, if you look at one of the parameters, the ADA1, which is a marker of cardiovascular disease, you can see that it's elevated in the women with gestational diabetes. And this is something you can do very easily using NMR. It's a 5-minute experimental runtime, and it gives you the information very quickly. So it's a very efficient way of looking at your blood lipids. Now, we can do things during pregnancy to change disease risks for ourselves and for our babies. And one of the things we can do is eat healthily. And a lot of women who are pregnant do eat healthily because they're aware of the influence of diet and living a healthy lifestyle on the baby. So this is just looking at some women who followed a Mediterranean diet, which is a healthy diet, versus women who didn't really bother with a healthy diet. And you can see just based on their food frequency questionnaires that at each of these time points during pregnancy, they are consistent. So people who said they were following a healthy Mediterranean diet had lower levels of dairy and sugars and things that we salts, things that we would consider bad. And just using the food frequency questionnaires, it seems that it's very consistent throughout pregnancy. Women who start off eating healthily tend to be the ones who continue to eat healthily all the way through pregnancy. So if you then take the urine and the plasma profiles and you look at whether you follow the healthy diet or you don't follow the healthy diet, you can see from the blue and red, the healthy and the non-healthy diet, that there are differences. And one of the big differences in the blood plasma profiles is a change in the glycoproteins, acetylated glycoproteins. We call this GLIC-A, GLIC-B. And these were 2 markers of inflammation that Jeremy actually discovered way back in the 1980s, I think. But now have become very popular markers of inflammation. And when people have compared GLIC-A against high sensitivity reactive CRP, which is our traditional blood measure of inflammation, it's found that the GLIC-A actually performs a little better in terms of the accuracy of predicting the inflammation. Now, the blood plasma, we're seeing changes in inflammatory process, so that the people who eat an unhealthier diet have a higher levels of these inflammatory markers in the blood. If we look at the urine, then what you see is that the people eating a healthy diet have increased levels of hip urate and gut microbial markers. And so we know that the microbiome is influenced by diet, and we know that the microbiome also is connected with inflammation, immunology, and health. So you can see very easily here that diet throughout pregnancy really affects your own profile and is obviously going to have some influence on that of the infant as well. So moving up the arrow a little towards getting older, one of the things that we find that is a big problem in particularly in developing countries, but also obviously in Australia, is burn injury. And a lot of children manage to get burn injuries. It's usually scalds from hot water or pulling pans off shelves. Now, I'm going to show you burn injury because it's something we've been working on, but you could really substitute any sort of childhood trauma like a traffic accident and so forth for most of the changes I'll show you. And this is work we've been doing with Fiona Wood and Mark Fear from the University of Western Australia. And Fiona is probably most famous. She was the person who developed spray on skin. So she's developed some sort of scaffold, skin scaffold that you can spray over burn in the cell to encourage cellular regrowth over the wound area. But if we just think of a normal burn, there are three different options. We can perform surgery and give skin grafts. There's drug treatments to things like antibiotics, for example, to prophylactically prevent infection, and other drugs to promote skin regeneration. And then we've got this spray on skin. And metabolically, we're taking 3 approaches. So firstly, we're going to use the eye knife that Jeremy explained, looking at cancer tumors, which trying to adapt that at the minute to use on skin. So how can you tell how much skin you've cut off somebody if it's viable? If you leave too much skin, it could later go necrotic and die, and it also contains toxins from the burn itself. If you take too much skin, then the bigger the wound or the surgical wound, the more likely you are to have post-surgery complications. The other things we're looking at is identifying the relationship between early intervention and downstream scar size. And thirdly, and perhaps most importantly, and this relates to other trauma, is if we can identify the mechanisms associated with increased mortality in burns patients, then we can really understand how trauma influences later life. So we've been doing some very preliminary work using animal models, but also looking at people who have burns, and we've got healthy skin and debrided skin or burnt skin. And if you try to look at the two types of skin, you can see in the scores plot on the left hand. Thank you. You can see up here, healthy skin and the burnt skin give a completely different profile, and it's all to do with lipids, particularly sphingomyelins. However, when we look at the skin sections from each person is colored by a different key here. The key is the color. You can see that every individual has a very particular skin chemistry, and there's one person in particular who had the burn is very, very different. So you can see there's a lot of work to do in terms of understanding into individual differences in skin chemistry and then relating that to burn versus non-burn. So digging deeper into defining the systemic, not the skin chemistry now, but the blood changes after burns, then we find a lot of biomarkers differentiating burns from non-burns, the healthy ones in green and burns in red. So this is on admission to hospital. You can see 6 weeks later, you still, even though some of these burn scars have healed, there's still a big difference. So although the skin is healing, you still see a metabolic signature, it still remains. And some of the parameters here I'll come back to the glycoproteins that I showed you before, the marker of inflammation, and also a new set of molecules that we've become interested in. Jeremy discovered this not so long ago, the supramolecular phospholipid cluster, and I'm sure he's going to say more tomorrow in the talk focusing on COVID, but it turns out that this phospholipid cluster is really important in the whole inflammation story as well. So this side of the slide is the NMR results. Over on the other side of the slide, we have mass spectrometry, and you can see again that as well as the NMR parameters, they're giving you one sort of information about mainly about the inflammatory markers. When you look at the mass spec, it's giving you a lot of information about sphingomyelins and detailed lipid species. So you can more or less tell how things are going to go when a patient comes in. Some patients respond very well to therapy, others don't. And one of the parameters, outcomes we use is, how long do you need to change dressings for? How long does a patient need a dressing for? And on admission, before you know whether you're going to need to change dressings very often, you can actually predict that from the very first time point. So based on the blood chemistry, you can see who is going to be a slower healer than the other burns. And again, this is coming down to some of the lipoproteins here. So your low density lipoprotein parameters in particular, which are the ones that we associate with more deleterious cardiovascular outcomes, for example, these are elevated in the patients who need further care and whose wounds don't heal correctly. So as I mentioned, this glycoprotein here, you can see this is on healthy patients. This is on admission to hospital, and you can see that the inflammatory parameters remain elevated for 6 weeks. And this is in adults, this population. Now I'm going to show you something that's a little more surprising, if you like. So if we look at the literature, and this is the Kaplan-Meier survival estimates, you can see that adjusting for confounders in children, so under 15 years old, burns patients, you have a 1.6 times greater mortality than people who have not been burned. So it doesn't matter if you don't have a scar, for example, even if you've healed completely, you still have higher mortality. And in adults, burn injury over 65 years of age, the average life expectancy drops from 12 years to 7 years. And it's mainly due to cardiovascular disease and malignancies. So when we're thinking of burns or even road traffic accidents or any other trauma, we're not just looking at the site of injury, we're looking at things like increased incidences of gastrointestinal diseases, you get muscle wasting, decreased bone density, and increased risk of all sorts of different infections. So one very small event in your life can have a big impact downstream. So what is beyond the burn? What is causing these chronic diseases? And what we can see, this is the NMR results showing burns in red and non-burn injury, children in blue. But these samples are taken 3 years after the burn. So these children are on the outside completely healthy. Most of them haven't got persisting scars, and they're just like any other child, except they have been changed metabolically. One of the things we can see, this is the mass spectrometry analysis and higher levels of amino acids. So we know that post-burn, people become hypermetabolic and have high levels of amino acids, also increased glucose, for example. But one of the things that was really interesting is if you look at these wheels, what they're showing is the correlation between different metabolites and different cytokines. So you can see that in the healthy compared to the burn here, that the burn children have far more connections between metabolites and cytokines. There's some sort of signature or some sort of interaction going on between the biochemicals in the blood and the immune parameters of cytokines like TNF-alpha and interferon gamma. And we see the same things if we look at the NMR data. And this again is just showing the difference between the burn here and the non-burn. And you can see quite clearly the elevated glucose, some of the elevated amino acids like glutamine and GLIC-A, the branched chain amino acids. So for example, branched chain amino acids elevations in plasma are linked, or are one of the things we see in type 2 diabetes. So again, this signature really looks like a very subtle signature of type 2 diabetes, except that these children are supposedly healthy and recovered from the burn. But again, you can see the huge amount of increase in the burn, the connections between the burn compared to the control. So we know that there's this handshake between the cytokines and the metabolites. And this is another instance. This is looking at parasitic infections, Leishmania major or cutaneous leishmaniasis. And this is a parasitic infection, infects the skin, and it gives very nasty lesions like this. Typically, they will resolve by themselves after 6 months. However, whilst the wound is open, obviously it predisposes to other infections, and the leishmaniasis is carried by the sandfly vector. It's high prevalence in Northern Africa and in China, for example. Now, this is a mouse model of leishmaniasis, and we compared a resistant strain to a non-resistant strain. So this is the non-healing model. This is the self-healing model. So 2 different strains of mice. And what you notice here again, these are all different urinary metabolites in yellow, blood in red. We've got some fecal metabolites in gray, and then some cytokines and gut microbes. But the overwhelming thing about difference between the non-healing model and the self-healing model is that there's a huge amount of intercorrelations in the self-healing model, and it's shown a little better in these graphs here. So the model that can heal itself is much better than actually talking. Different parts of the body are very much better talking to each other. So the cytokines are initially raised very quickly, and they are related. They have a correlation with a lot of the urine and plasma metabolites. So you see this immunometabolic signature comes out very strongly. So you need this to be able to heal. So in an acute lesion or acute trauma, then being able to heal, having this rapid immunological and metabolic response, co-response, is a really good thing. However, it's good for an acute situation. But if you continue on through life and you still have this immunometabolic interaction, this then is becoming not so good for your health downstream. And it's associated with inflammation, which is one of the things that we worry about in diseases like cardiovascular disease. And I'll show you another example of this. This is a comparison of parasitic infections that Jir and Yasmin and I did a long time ago. But we had parasites like protozoa, malaria, African sleeping sickness that are predominantly Th1. So protozoan, very strong acute infections that really need a strong response from the body to clear them. And then you have other parasitic infections like helminths, worms, Fasciola hepatica, and Schistosoma mannsoni, and liver fluke, for example. And these don't really cause this massive immune response because they need to stay hidden from the body, because the longer they stay hidden, the more they can grow. And these tend to be the ones that are slower burning but do damage in the long term. So you've got a Th1, classic Th1 versus Th2 immunological difference. And we can see here that the Th1, predominantly the protozoans are here, whilst the liver flukes, the Th2 immunological parasites, are up here. And so we see this metabolic difference predominantly driven by whether it's a strong acute response or going on to the chronic immunological response. And again, we see these cytokines that are strongly linked to some of the metabolites that we're detecting. So coming back to one of my diagrams, and now looking at the impact of all these early and acute exposures on chronic diseases, we're going to focus on the later life chronic diseases for the last part of the talk. So one of the real problems in the UK is dementia, basically. We have an aging population, lots of people getting Alzheimer's disease and Parkinson's disease. And we know that some of this is linked to different loci in the genes. So we have different SNPs or polymorphisms, and these were found from the airwave study conducted by Paul Elliott on 500,000 policemen. So we're interested particularly in the CH18 SNP, and this work has just recently been published in PNAS. So what we did was we ran metabolic profiles using mass spectrometry and NMR, and we ran them at the London National Phenome Centre. And we regressed the metabolic profiles against the SNPs. And it turns out that a lot of the SNPs are linked to or highly correlated with lactosyl or hexosyl ceramides. And across the different mass spectrometry assays, we see some very strong correlations with these ceramides. So the SNP is most closely related to the ABCA7. So we took an ABCA7 knockout mouse model just to check out the ceramide profile. And what you find is that you can very clearly see the difference between the knockout and the wild type. And if you look at the ceramides between the wild type and the knockouts, they're highly elevated in the knockouts, mainly in the males, but not in the females. So we can see, we can take our population metabonomics, and we can take our population genomics, and we can run it through a mouse model, and we can validate that these ceramides are indeed related to the Alzheimer's phenotype. Now, finishing with some examples in liver cancer, I know that liver cancer, particularly cholangiocarcinoma, is a big problem in Kaen and the surrounding area. And I want to start off, though, with a different type of liver cancer. This is hepatocellular carcinoma, which is the major killer in West Africa, and also one of our biggest liver cancer problems in the UK. This was work done by Abelona Mueang, and she was one of the PhD students at Imperial. And what she did was she started off with a literature search on metabolic phenotyping, and she found, I think it was about 83 different studies out there. And she looked at these and went through them. And then as she was going through them, she noticed that actually, if you took these 80 studies, a lot of them didn't have a good comparison. So they were comparing the cancer with healthy controls, when actually you probably need to compare it with cirrhosis, because the liver cancer develops against a background of cirrhosis. So half of the studies had no control, appropriate control. Some of the studies had only 5 or 6 samples in. So what she did was to develop a scale for weighting the literature. And she came up with some of the groups of metabolites that seem more robust, those mainly being acyl carnitines and bile acids. So these were the ones from the literature that we focused on going forward. So you must be careful when you look in the literature, when you see metabolic markers, people doing metabolic phenotyping, you need to check the experimental conditions and the statistics, because a lot of the literature is not the best. So you've got to use some judgment in deciding whether you follow their methods or not. So as I said, hepatocellular carcinoma, big problem in the Gambia, is the biggest killer in males under 40. And we had a very nice labs at Imperial, as you've seen. But what we really wanted to do was make the technology available for people in West Africa, because it's no good them sending the samples to us. So what we were trying to do was use our high-tech technology to identify biomarkers in the UK and then transfer that back to Africa. This is the setting in the clinic in West Africa. It's it's resource limited. They don't have a lot of money, but Waters very kindly donated a mass spectrometer, very robust, simple mass spectrometer. So our methods had to be simple. And we looked at the sample population, and we identified biomarkers in the UK and said, well, let's see if we can develop something that we can run in Africa. So the strategy was to collect samples in the Gambia, develop a method, simplify it, and then reintroduce it to the lab in the Gambia. So these were Nimzing Ledep's work, and you can see that when you look at different comparisons here, the green is the cancer, and the blue is the yellow is healthy. You can get a nice separation, a really strong model here. Similarly with other liver diseases. But if you look at this last map here, this is compared against cirrhosis. So it's not so easy to detect hepatocellular carcinoma against cirrhosis. And if you look at some of the discriminatory metabolites, things like methionine, N-acetyl glutamate, you can see that it goes from healthy. You can easily see the difference between the hepatocellular carcinoma and the healthy, but cirrhosis also has a lot of these biomarkers too. So how do we really address that? And if you look at stage of cancer, you can see that these markers from the NMR, the acetyl carnitines, the N-acetyl glycoproteins, they increase in concentration in the blood as the cancer stage gets worse. So we looked at some targeted mass spec assays. things like carnitines and acyl carnitines. Again, you can see that, yes, there's a big difference between control and cirrhosis, control and the cancer, but also the cirrhosis is halfway in between. So what is cirrhosis and what is the cancer? So it's very clear you can't just use one or two of these markers, because you're not going to detect the cancer with any sense of specificity. But it still outperforms the classic clinical gold standard test, which is the alpha feta protein. Bile acids is another set of metabolites that are associated with the cancer. Again, even more so here, you can see that there's a big difference between the healthy and the cancer, but that you can't really differentiate the cirrhosis. However, if you take 3 of the top discriminatory metabolites, which is propionyl carnitine, indole acetate, and glycolic acid, which is a bile acid, and you use these 3 discriminatory markers, 1 acyl carnitine, 1 bile acid, and 1 gut microbial metabolite, you can see here now we are starting to see with some degree of specificity. So this is a strong reason for why you need a panel as opposed to a panel of metabolites as opposed to a single marker. And then moving on finally to some of the cholangiocarcinoma, which is very relevant to the Kaen area. If you compare some of the metabolites again with different liver diseases and the cholangiocarcinoma, again, you can see you can get some discriminatory patterns, but a lot of these metabolites, it's worth noting, are coming from things like phenylacetylglutamine in the urine, indoxyl sulfate. These are showing a perturbation in the gut microbiome. So whether the microbial changes are linked to the disease causally, or whether they're a consequence of the disease, that is the part that we don't know yet and we need to look at. But regardless of whether they're causal or consequential, perhaps they can be useful as a biomarker of disease presence and disease stage. And again, you can see some of the stripped out assays like acetyl carnitine and purine metabolism. So if you know what you're looking for, you can optimize your methods to measure and quantify these metabolites. So very final slide is just really to bring you back to the idea at first, where we have different states in life, whether it's pregnancy journey, whether it's having a fatty liver through bad diet, being obese, or maybe having an infection like COVID or hepatitis. All of these events cause changes in inflammation. So they set up or they heighten the inflammation. The inflammation causes changes in things like cytokines and also the relationship between cytokines and systemic metabolites. And these changes impact our various organs. So you can move towards diabetes or heart disease, neurodegeneration. But the inflammatory signature underlying these early on appears to be very similar. So one of the big questions is, how do we use our technology to tell what sort of disease you might be at more risk of? And this is where we look at the molecular dysregulation. We can look at things like neurotoxic metabolites. We can look at branched chain amino acids and metabolites, for example, linked with the development of type 2 diabetes, or the lipoproteins associated with cardiovascular disease. So using our suite of metabolic tools, we can try and profile a wide range of metabolites to give us information as to what we are more at risk of. And this is all to do with the allostatic load and maintaining our balance on the side of life, rather than when our bodies give up on homeostasis and move towards sort of an irreversible trajectory into disease. So with that, I'd just like to thank the team at the Australian National Finance Centre, and also our collaborators at Imperial and other universities around the world. And the liver clinicians, mainly from Imperial. So thank you for your attention.
Thank you, Professor Ellen Holmes, for this great talks and allow us to see these huge applications of the metabolomics and phenomics approach to a varieties of medical questions and problems. So any questions from the audience? Just one quick question. Okay, I have something very brief, just interest of time. I saw you work on a huge range of research questions and also with the researchers of a wide range of backgrounds. So could you just tell us what is your approach for trainings, next generation of scientists into the fields of the phenomics? When you work with a huge range of researchers and scientific and scientists from the various backgrounds, and what would be your approach in training them to adopt the phenomic approach into their research?
I think one of the key things is to spend time in a lab that's routinely running the phenomics and really learn what sort of quality of samples you need, starting from the actual sample collection, but right the way through the analytical technology. And then importantly also on the statistics side, how to know when your experiment's valid, how many samples do you need, how do you know you're not overfitting models, how to validate. I think just being able to follow the process from start, it doesn't matter what you're going to focus on as an analytical chemist or statistician, but actually seeing the process from start to finish and understanding it, I think, is important. And
allowing them the time to go through those processes. Thank you so much for your inspiring talks and for the showing us all this application. Please give Ellen Holmes the big round of applause. Thank you so much. And for our next speakers,